Yes, good evening. Good evening, everyone. Dr. Pranjal, Ash, Harsh, Chirag, Usman, good evening. Good evening, beta. I hope the audio and visuals are perfectly fine. So before starting the session, just give me a thumbs up in the chat box. Yeah, that everything is fine so that we can start, okay, or we can continue with our biochemistry session. Yes, Jainam, good evening, beta. Flight Education Center, good evening. Okay, Faha, good evening, beta. Good evening. Yes, good evening. I hope everything is fine. Yes, Chirag says everything fine. Yeah, flight. Okay, Jainam, very good, beta. Perfect, perfect. So let us just start with the session. And today we are going to discuss about the glycogen metabolism. Today we are going to discuss in detail about the glycogen metabolism. Yes, in detail about the glycogen metabolism. So let us start. Okay, let us start. So let us okay understand few things regarding glycogen first. Now, what is glycogen, beta? Glycogen. Already I have just discussed this, but as you all know, I always start from the basics, beta. That's why you should know that glycogen, it is the storage form of, yes, it is the, yes, okay, perfect, perfect, beta. Yeah. So remember, beta, the glycogen, it is the storage form of glucose. It is the storage form of glucose in our body, right? It is the storage form of glucose in our body, right? Now you already know that the glycogen, beta, glycogen, it is a polymer of glucose, right? It is a polymer. It is a polymer of glucose. Now this polymer of glucose, you already know it is a branched polymer. Okay, it is a branched polymer. So everyone knows that suppose this is the structure of the glycogen. Okay, suppose this is the structure of the glycogen. Now, glycogen, it is a polymer of glucose. So I have joined glucose like this, right? I have joined glucose like this. Okay, this is a linear chain. It is a linear chain. Okay, this is a linear chain. Remember, beta, the glucose will also be present at branching. Okay, it will be also present at branching points, right? So, yes. So, this is something which is called as the glycogen. It is the structure of a glycogen. Okay, it is the structure of a glycogen. Remember, beta, between glucose and glucose in a linear chain, beta, in a linear chain between glucose and glucose, one bond is present, beta, and remember that is called as the alpha 1,4 glycosidic bond. I have already discussed this. It is alpha 1,4 glycosidic bond. Okay, it is alpha 1,4 glycosidic bond. This I think everyone knows. I hope this everyone knows. Apart from that, beta, apart from that, the bond which is present at the branching point, okay, only at the branching point, beta, it is alpha 1 to 6, alpha 1 to 6 glycosidic bond, alpha 1 to 6 glycosidic bond, that you already know that, yeah. The carbon atom of glucose 1, I mean the carbon of glucose, no, the glucose carbon atom 1 will actually bond to the another glucose carbon atom 6. Okay, and here it is between the first carbon and the fourth carbon. Okay, so that is something that you already know. Even over here also you will get, okay, alpha 1,4 glycosidic bonds. Only at the branching point, beta, it is alpha 1 to 6 glycosidic bond. Only at the branching point it is alpha 1 to 6 glycosidic bond, right? So yes, that is something you have to remember. Apart from that, beta, you have to remember two more points regarding glycogen. Regarding glycogen, yeah. Remember, beta, that glycogen. Okay. Now I have a question to everyone in the chat box over here. I have a question to everyone over here that beta, we have two glycogen is found in the two parts of the body. One is the muscles and one is the liver. Now, which is more beta, muscle or liver content? You already know which is more. I hope everyone knows that the muscle mass is more as compared to the liver. So obviously the glycogen, okay, it will be more in case of what, liver or muscle. Type in the chat box beta. Yes, glycogen किस में ज्यादा होगा, मसल में ज्यादा होगा या लिवर में ज्यादा होगा? आपको पता है liver is a gland, right? And muscle is present, the skeletal muscles are throughout present in our body, right? So you already know beta, you already know yeah, you already know we have storage of glycogen, we have storage of glycogen in the, yes, two sides. Okay, we have two sides. One, it is the liver. Okay, and second, yes, everyone is right beta. Chirag, Jainam, Usman, Ash, I will come to your doubt, Fahad. Yes, Harsh. Okay, very good. And second, it is the muscle, and second it is the muscle bit. Now muscle, basically you already know that muscle is more in the body as compared to liver. So when I say the muscle, okay, it contains the highest, okay, it contains the highest total body content. बॉडी में सबसे ज्यादा जो ग्लाइकोजन है वो मसल में देखने को मिलता है. ओके, इट इज द हाईएस्ट टोटल बॉडी कंटेंट of glycogen in the muscle, glycogen in the muscle, right? Apart from that beta, if we talk about the liver, if we talk about the liver, if we talk about the liver, it is the highest percentage by tissue weight, by tissue weight. Okay, so it contains the highest percentage of glucose. It is by the tissue weight as compared to the muscles. Liver is having like the highest percentage of glycogen by the tissue weight. Overall, the glycogen it is more in the muscle. Why? Because muscles are more as compared to like the liver. Liver is only one, right? And if we talk about specifically about the tissue weight, yes, liver is having more glycogen, okay, as compared to this. So I hope this is clear to each and everyone. Yeah, I hope this is clear to each and everyone. So yes, just give me a thumbs up in the chat box, beta, if this basic thing is clear to everyone. If this basic thing is clear to everyone, right? Chaliye. Chaliye, Harsh, very good, beta. Harsh, very good. What about rest, beta? Usman, very good. Very good, beta. Jainam, very good. Yes. I have already told that they are, yeah, stored in a form of particle, in the form of particles. Can anyone name the particle? Yes, yes, Chirag Lalwani, very good. Ash, very good, beta. Yes, Jainam, very good, very good. Now let us now discuss the glycogen metabolism. Now let us now discuss about the glycogen metabolism. Glycogen metabolism. Yes, you already know, guys, you are already aware of the fact that, yeah, when we are consuming carbohydrates, okay, when we are consuming carbohydrates, you are very well aware with the fact that, yeah, the carbohydrates that we are consuming, the carbohydrates that we are consuming, it is mainly in the form of disaccharides, right? It is mainly in the form of disaccharides, and these disaccharides will go into the stomach and will break down into glucose. Okay, will break down into glucose, right? Will break down into glucose. Now, from the stomach, beta, you already know we have one organ, okay, that is called as the liver. Okay, You already know we have this liver, and from the stomach, this glucose will reach the liver. Okay, this glucose will reach the liver from the stomach. Okay, through the portal system. Okay, through the portal system, it will try to reach the liver. Okay, through the portal veins. Yeah, this glucose will be absorbed, and this glucose will be sent to the liver. This glucose will be sent to the liver, right? So you already know that this glucose, okay, this is the portal system, okay, through which this glucose is entering into the liver. Okay, and now this glucose, okay, now there is so much of glucose beta. What will happen to this so much of glucose? These glucose will combine with each other. Okay, this glucose will combine, okay, with each other to form glycogen, and this is something which is occurring in the liver, and you know the process. Glycogen is formed beta, so anything which is formed, it is genesis. So it is glycogenesis. It is glycogenesis, right? Now when there is too much of glucose in the body, okay, when there is too much of glucose in the body, you already know during fed state. Now, when I say fed state beta, you are already aware that I am talking about 1 to 4 hours after food. 1 to 4 hours after food. And this is the state where beta, yes, this is the state where in the body there is so much of glucose. When you have just taken your diet, imagine beta, when you have just eaten or taken your diet, what will happen in the blood? Okay, in the blood, the glucose levels will rise. Now this blood glucose will go into the liver, and some glucose will be transferred to provide energy, whereas some glucose beta, okay, it will be converted into glycogen at storage, okay, for storing. Okay, so when I say glycogenesis beta, it will occur in which state? It will occur in which state beta? ग्लाइकोजेनेसिस बेटा, जब ग्लूकोज का लेवल ज्यादा होगा तभी तो ग्लाइकोजन बनेगा, तभी तो वो स्टोर होगा. So obviously it is always in the fed state beta, fed state. Now imagine beta, what happens after some time? Okay, like, yeah, we have a state of early fasting when you have not taken any. Very good, Usman, very good beta. It is the fed state, early fasting. Now when I say like 4 to 16 hours, 4 to 16 hours, okay, after food, you already know you are not taking any diet. You are not taking any diet, but yeah, now the body needs what we say, glucose. Now the body needs glucose. So how this glucose will come? This glycogen will again break down into glucose, क्योंकि पहले तो ये स्टोर हो गया था. अब क्योंकि अर्ली फास्टिंग में बेटा, अर्ली फास्टिंग में यू आर नॉट टेकिंग एनी डाइट, but your body needs glucose, your body needs ATP. So this glucose will come from glycogen. Now this glycogen will break down into glucose, and that is something which is called as glycogenolysis. This is something which is called as glycogenolysis. Glycogenolysis का मतलब क्या है? Glycogen, lysis means breakdown. Lysis means breakdown. So if we say glycogen metabolism beta, if we say glycogen metabolism, yeah, glycogen metabolism. Now it is of two types beta. Yes, from yeah, we have glycogenesis, we have glycogenesis, and we have glycogenolysis. Glycogenolysis. Glycogenesis basically means formation of, yes, formation of glycogen, okay, from glucose, formation of glycogen from glucose. You already know glucose will be getting converted into glycogen. This is like we have to learn how does this happen. This will be our first topic for today, that is glycogenesis. And glycogenolysis means Glycogen is broken down. Glycogen, it is broken down. Okay, lysis means broken down. Okay, to glucose. To glucose. So you already know this is exactly opposite, glycogen to glucose. Now, glycogenesis basically occurs in the fed state when you have taken the meal beta. Okay, when there is excess glucose in the blood, okay, at that time the glycogenesis will take place. But when I say glycogenolysis, it is in the state of early fasting beta. Early fasting, when you are not consuming any diet and your body needs glucose for energy. Okay, so that is that is the early fasting. Yes, it is glycogenolysis. This glycogen will break and it will try to replenish. Okay, it will try to meet the requirements of the body. Okay, the needs of the body. Okay, by providing glucose. Yes, yes, anabolism and catabolism very good for her. Yes, you already know जब glucose से glycogen बन रहा है. Yes. So I hope this is clear to each and everyone over here. I hope this is clear to each and everyone over here. Just give me a thumbs up in the chat box, beta, if you have understood the basic concept that what we are going to study in the glycogen metabolism. That what we are going to study in the glycogen metabolism. Yes, Janam, very good. Chirag, Harsh, very good, beta. Very good. Perfect. Perfect, beta. So we have just discussed about Usman, very good. We have just discussed about the some points related to glycogen and the glycogen metabolism overview, like what we have to like discuss. Now, the very first thing that we will discuss, it is glycogenesis. Okay, it is glycogenesis. Glycogenesis. And when I say glycogenesis means you already know it is glycogen synthesis, right? It is glycogen synthesis. Yes, it is glycogen synthesis. Now, my question to everyone over here in the chat box that why glucose it is not stored in our body as it is? Why glucose needs to convert into glycogen before storing? Have you ever thought about this, beta? Just tell me in the chat box if anyone knows about this thing. So you already know when we say, yeah, glycogen, okay, it is storage form of, it is storage form of carbohydrate, right? Or yes, the glucose, carbohydrates or the glucose beta, right? Now you already know beta. If now let us suppose beta, if glucose it is stored inside the cell, if glucose it is stored inside the cell beta, okay, सबसे पहली बात this glucose will take a lot of space, right? Now glucose it is water soluble beta. Yes, हाँ ठीक है. हाँ, चलो देखो अभी इसका रीजन देखते हैं जैना. ग्लूकोज बेटा बेसिकली विल टेक अ लॉट ऑफ स्पेस एज कंपेयर टू ग्लाइकोजन. ग्लाइकोजन मैंने बताया था बेटा ब्रांच्ड पॉलीमर है, ब्रांच्ड पॉलीमर है. तो ऑब्वियसली बात है उसका सरफेस एरिया ग्लाइकोजन का कम होगा एज कंपेयर टू ग्लूकोज. ग्लूकोज ग्लूकोज अलग अलग पड़े रहेंगे तो हैव दे विल ऑक्यूपाई अ लॉट ऑफ स्पेस. So generally thinking beta, if glucose is stored inside the cell, if glucose is stored inside the cell, what will happen? It will consume a lot of space. Secondly, the glucose is water soluble. Okay. Now, water soluble glucose, what it will do? It will absorb. Okay. It will absorb glucose. It will absorb glucose from extracellular matrix. From extracellular matrix. And yes, extracellular matrix. Yes. And what will happen? It will take all the glucose inside and the cell will be swelled up. Cell will be swelled up. Okay. For example, this is the cell. Okay, this is the cell, and in the cell we have glucose. So glucose will take all the water inside, and this will result in cell swelling. This will result in the cell swelling. This will result in the cell swelling. So yes, that's why no organ can store glucose. Okay, that's why no organ can store glucose. Now, beta, on the other hand, if body, okay, if body needs glucose, needs glucose, so you already know glycogenolysis, glycogenolysis, it can take place at any time. Okay, it can take place at any branching point. It can take place at any branching point, and glucose releasing from the branching point will be very fast. Glucose releasing from branching point is very fast. Okay, branching point is very fast. So whenever body needs glucose beta, what will happen? Glycogenolysis will take place at that particular time. And you already know glycogen, it is a branched polymer. So yeah, at the branching point, from the branching points, the glucose will be start releasing. And that releasing from the branching point, it is very, very fast. So that is one of the reason that it is stored as glycogen. That is one of the reason that it is stored as glycogen. Yes. And remember, beta, glycogenolysis, okay, can take place. Yes, it can take place in liver, it can take place in muscle cells, and glucose can be immediately released. Okay, so that's why it is stored as glycogen in most of the yeah in most of the yeah it is stored in the like it is stored as glycogen. Okay, so I hope this concept is clear to you, why glucose cannot be stored and why glycogen is used for storage. Okay, so just give me a thumbs up बेटा, अगर दिमाग में ये कंसेप्ट क्लियर है बेटा, रट्टाफिकेशन नहीं लगाना, आपको कंसेप्चुअल चीजें समझनी है. आई एम वेरी फोकसिंग ऑन द कंसेप्ट्स बेटा, आई एम टेकिंग टाइम टू एक्सप्लेन यू द कंसेप्ट, सिर्फ रिएक्शंस याद नहीं करनी होती हैं. सोचो इसको मेडिकल साइंस से वी हैव टू लिंक दिस बेटा, we have to link this with the medical clinical part, right? Why do we need this glycogenesis, right? Why do we need this? Yes, Ash, Faha, Harsh, Chirag, very good beta. Chalo, let us now focus on glycogenesis now. You guys will tell me glycogenesis takes place in which state, fed state or fasting state, and under the influence of which hormone, and under the influence of which hormone. जब बॉडी में ग्लूकोज ज्यादा होता है तब कौन सा हार्मोन एक्टिवेट होता है? Whenever the blood glucose level is high, which hormone gets activated? You guys are going to tell me. Yes, yes, Chainam, very good beta. Yes, Chainam, very good beta, it is the fed state and the insulin. Yes, so you already know beta, if we say yeah, if we say glycogen synthesis, yes, very good beta, very good, and it is taking place in the fed state. Yes, Harsh, Usman, very good. Harsh, Chirag, very good beta, it is the fed state beta, it is the fed state. And remember, it is taking place under the influence of insulin hormone. Insulin hormone, it is released by which cells? Okay, insulin hormone, it is released by which cells of the pancreas? I want every one of you to answer this question. Yeah, all the first years. Yes. So, yes. Yes, very good Varun Kashyap, very good Chirag Lalwani. It is the beta cells of the pancreas. Yeah, it is the beta cells of the pancreas which is responsible for the secretion of the insulin. Very good Usman, very good Chirag, very good. Chal, chal. Very good beta. Now you already know. Let us suppose this is the cell. Okay, this is the cell. I will be discussing the reactions also, but before that, just try to focus over here, beta. Now this is the cell and this is the cell membrane. Okay, this is the cell membrane. I'm always trying to show you a line diagram. What is happening, beta? You already know that we have one transporter, and that is GLUT transporter, and specifically GLUT1. It is present throughout. So yes, we have GLUT1 transporter, which is associated with the transfer or movement of glucose from outside to inside the cell. From outside to inside the cell. Yes. बेटा फास्टिंग स्टेट में कैसे होगा? आप खुद ही सोचो कि बॉडी में जब ग्लूकोज है ही नहीं, फहा बॉडी में व्हेन द ग्लूकोज इज नॉट देयर, फास्टिंग में क्या होता है? बॉडी में एनर्जी की जरूरत है ना, ग्लूकोज है ही नहीं बॉडी में, तो ग्लूकोज जो बचा हुआ है बॉडी में वो एनर्जी प्रोवाइड करेगा या ग्लाइकोजन बनाएगा? कॉमन सेंस है कि अगर फास्टिंग की स्टेट है तो ग्लूकोज तो सारा एटीपी प्रोड्यूस करने के लिए यूज होगा ना, एरोबिक ग्लाइकोलिसिस होगी, राइट बेटा? ग्लूकोज तभी ग्लाइकोजन बना सकता है जब ब्लड में ग्लूकोज ज्यादा है, और ब्लड में ग्लूकोज तभी ज्यादा होता है जब आपने तुरंत खाना खाया हो, व्हेन यू हैव टेकन योर डाइट, ओके, एट दैट टाइम ओनली द ब्लड ग्लूकोज लेवल इज हाई। फास्टिंग में तो ब्लड ग्लूकोज लेवल हाई नहीं होता ना बेटा, ठीक है? चलिए, ये सलाद वेरी गुड बेटा। चलो 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 चलो, नाउ remember beta, now this glucose inside the cell will get converted into glucose 6 phosphate। देखो बेटे, पहले बेटा ग्लूकोज था, ग्लूकोज में 6 कार्बन होते हैं, और ग्लूकोज 6 फॉस्फेट में क्या हो जाएगा? 6 कार्बन पे फॉस्फेट ग्रुप लग जाएगा। नाउ दिस ग्लूकोज 6 फॉस्फेट बेटा, this glucose 6 phosphate, yes, it has two fates beta, okay, it has two fates. Either it can go for glycolysis, either it can go to glycolysis, और glycolysis में जाएगा तो एटीपी बनाएगा, एनर्जी बनाएगा, और it can go to, it can undergo glycogenesis, glycogenesis, basically to form glycogen, to form glycogen. And remember beta, whenever the blood glucose level is high, whenever the blood glucose level is high, glycogenesis is taken as priority, okay, more than in well fed state. If you just want me to conclude it in a statement, in well fed state, आपको भी पता है पहली रिएक्शन क्या होती है ग्लाइकोलिसिस में? ग्लूकोज से ग्लूकोज 6 फॉस्फेट बनता है। You already know, I have already talked about this in detail beta. We are starting with the glucose. Yeah, we are starting with the glucose, and from the glucose what is happening? Yeah, glucose 6 phosphate is formed. Now glucose 6 phosphate can undergo glycolysis, or it can undergo glycogenogenesis also. Now if we talk about in well fed state beta, in well fed state, what will happen? Yes, what will happen? This glucose 6 phosphate, okay, will undergo glycogen synthesis, glycogen synthesis more than glycolysis, more than glycolysis, more than glycolysis. So that is something that you have to remember, or the takeaway point over here is कि जब भी वेल फेड स्टेट होता है, वेल फेड स्टेट होता है आपने तुरंत खाना खाया है, ओके, 1 to 4 hours और 0 to 4 hours after food, till 4 hours बेटा, till 4 hours बॉडी ग्लूकोज को स्टोर करना चाहती है, एंड दैट इज समथिंग व्हिच इज मोर डन, दैट इज ग्लूकोज को स्टोर करना है, तो दैट इज कॉल्ड एज ग्लाइकोजन सिंथेसिस, ओके, इट इज अकरिंग मोर than glycolysis to provide energy, right? So we have now, I hope this is clear to everyone over here. I hope this is clear to everyone over here कि body में glycogenesis जो है well fed state में होता है, और priority level पर glycogen synthesis is more in well fed state as compared to glycolysis, right? So I hope this is the particular point, it is clear to everyone. Harsh Chirag, very good beta. Chalo. Now you already know the site beta. If we talk about the site, Chalo, Chirag Usman, very good, beta. You already know the site where it is happening. So remember, beta, it is, yeah, it is the liver and muscle. So we have liver and muscle, the two sites where it is happening. Okay, the two sites where it is happening. And organelle beta, remember it is cytoplasm. Paha beta, I've taught gluconeogenesis in the previous lecture. Beta, if you are a fake ID and if you are trying to manipulate my mental peace, please leave my session as of now. Don't try to disrupt me when I'm teaching. Please, all the students who are watching the lecture, kindly ignore the comments that is given by Faha, right? So, yes, sure. I hope the site is clear and the organelle beta, it is the cytoplasm. It is the cytoplasm beta, right? So, yes. Now, starting with the steps. So, we are discussing glycogenesis and we have three steps beta. We have three steps in glycogenesis. The first step beta, it is synthesis of, okay, it is synthesis of UDP glucose. I will come to one by one each step. Synthesis of UDP glucose. The second one, it is the reaction of glycogen synthase. Glycogen synthase. And the third one, beta, it is the step of branching. Step of branching. So we have to discuss each step one by one. We have to discuss each step one by one. Now, beta, the first step, synthesis of UDP glucose. Okay, synthesis of UDP glucose. Now, before coming on to the first step, there are 2 points that you have to remember over here. There are 2 points that you have to remember over here. The first point, beta, yes, glycogen is always synthesized on a primer. Okay, it is always synthesized on a primer, right? It is always synthesized on a primer, right? Now, when I say primer, okay, primer over here, it is again an important question for the exam. It is glycogenin. It is glycogenin. Now, what is glycogenin? Now, what is glycogenin? Now, beta, before coming to this, why there is a need of primer? Now, remember, beta, there is an enzyme that is called as glycogen synthase. There is an enzyme which is called as glycogen synthase. Now, glycogen synthase can only add glucose, okay, on a previously synthesized glycogen, okay, or a part of it, okay. So yes, it cannot add glucose directly, beta, okay, it cannot add glucose directly, okay. So that is why it needs a primer, and that primer it is called as glycogenin, and that primer it is called as glycogenin. Now when I say glycogenin, beta, remember it is a पॉलीपेप्टाइड पॉलीपेप्टाइड कहने का मतलब है ये प्रोटीन है. It is a protein. Poly means many, peptide means peptide bond, and this polypeptide, it is simply protein. So glycogenin is a protein, okay, which contains lot of which contains lot of tyrosine residues, which contains lot of tyrosine residues, okay. So yes, tyrosine residues, right? So that is something that you have to remember in the beginning only, that glycogen is always synthesized on a primer, and that primer over here, it is glycogenin, and this glycogenin is nothing but a polypeptide. Okay, why the glycogen always needs a primer? Because glycogen synthase is an enzyme, okay, which will help in the synthesis. It cannot add glucose directly, okay, so that's why it needs something to begin from, और beginning के लिए उसको एक primer की जरूरत होती है, and that is something which is called as glycogenin, and yes, which contains lot of tyrosine residues. That is again an important question. That is again an important question. Now coming on to the first step, beta. Coming on to the first step, that is synthesis of UDP glucose. Synthesis of UDP glucose. Synthesis of UDP glucose. Now, you guys will tell me. Now, let us start from the very basics. Let us start from very basics. You already know we have glucose, and glucose से अब हमें क्या बनाना है? Glycogen. So you already know the first reaction. The first reaction will be the glucose will be converted to glucose 6 phosphate. Now you guys are going to tell me which enzyme will be used, because this reaction I have discussed multiple number of times. Which enzyme will be used, and which ion will be used? Which ion will be used? Yes, and this phosphate will come from where? And this phosphate will come from where? Yes, Chirag Lalwali hexokinase है, हर्ष hexokinase है, very good, जयनाम very good. चलो, ये बात तो सबको क्लियर हो गई कि ग्लूकोज से ग्लूकोज 6 फॉस्फेट बनेगा. देयर इज अ नीड ऑफ एन एंजाइम ओवर हियर, दैट इज हेक्सोकाइनेस. हेक्सोकाइनेस, राइट, काइनेस. यस, यूज हो गया. Yes, everyone is right. Chirag Lilwani, Mg2 plus, Chenam ATP, Usman, very good. Usman, very good. Very good. Chirag, very good, beta. So you already know what will happen. One ATP molecule, okay, one ATP molecule, adenosine triphosphate, will break into adenosine diphosphate, adenosine diphosphate. And yes, ट्राई से 3 से क्या हो गया? डाई, 2 हो गया. तो एक फॉस्फेट कहाँ चला जाएगा? ग्लूकोज पर. ग्लूकोज यू ऑलरेडी नो इट इज अ 6 कार्बन कंपाउंड, राइट. ग्लूकोज का फॉर्मूला क्या होता है? C6H12O6. तो इट कंटेन 6 कार्बन. ग्लूकोज 6 फॉस्फेट में भी क्या होगा? 6 कार्बन होंगे. बट नाउ व्हाट विल हैपन? द 6 कार्बन विल कंटेन फॉस्फेट ग्रुप, और ये फॉस्फेट कहां से आया है? एटीपी से. तो जब भी okay, always remember beta, whenever there is a transfer of phosphate group from one molecule to another, we use an enzyme that is called as kinase. We use an enzyme that is called as kinase, right? एटीपी एक मॉलिक्यूल है, ग्लूकोज एक मॉलिक्यूल है. एटीपी से ग्लूकोज में फॉस्फेट ट्रांसफर हुआ है. ट्रांसफर इज for transfer of phosphate from one molecule to another, we use kinase. Hexo means 6 carbon compound. Hexo means 6 carbon compound. So just the name of the enzyme, hexokinase. Yes, everyone is right. Chirag has already given, Harsh has already given the answer. It is Mg2 plus. It is Mg2 plus. That is required as an ion. So up till here, I suppose everyone is clear. Yes, everyone is like clear in their mind what is happening. Now from glucose 6 phosphate beta, from glucose 6 phosphate, what will happen? From glucose 6 phosphate, and yeah, you guys are going to tell me a compound will be formed that is called as glucose 1 phosphate. Tell me the name of the enzyme beta. Tell me the name of the enzyme. अब चलो मैं स्ट्रक्चर बना देता हूं. अब कुछ नहीं हुआ है. या यू ऑलरेडी नो. यस, पहले खुद ही देखो. पहले 6th प्लेस पे कार्बन था, सॉरी 6th प्लेस पे फॉस्फेट था. यस, 6th प्लेस पे फॉस्फेट था, और अब फर्स्ट प्लेस पे फॉस्फेट आ गया. फर्स्ट प्लेस पे फॉस्फेट आ गया. ट्रांसफर ऑफ फॉस्फेट ग्रुप फ्रॉम वन पोजीशन टू अनदर पोजीशन इन द सेम मॉलिक्यूल. नहीं बेटा, आइसोम ठीक है, आइसोमराइजेशन रिएक्शन है. आई एग्री विद द फैक्ट, बट एंजाइम का नाम तो बताओ, ये आइसोमेराइजेशन रिएक्शन है, आई एग्री विद द फैक्ट, बट व्हाट इज द नेम ऑफ द एंजाइम बेटा, व्हाट इज द नेम ऑफ द एंजाइम? एक ही मॉलिक्यूल में जब फॉस्फेट ग्रुप ट्रांसफर हो रहा है फ्रॉम वन पोजीशन टू अनदर, so the enzyme that we use beta over here, it is mutase, it is mutase, और ये मुझे बताना भी नहीं चाहिए तुम लोगों को, so yes it is phospho glucomutase, it is phosphoglucomutase। म्यूटेस मैंने तुम लोगों को पहले भी बताया था बेटा, म्यूटेस मैंने तुम लोगों को पहले भी बताया था कि whenever there is a transfer of phosphate group in the same molecule from one position to another, तो ग्लूकोज 6 फॉस्फेट से क्या बन गया, ग्लूकोज 1 फॉस्फेट, ग्लूकोज 1 फॉस्फेट, तो उसमें हम लोग म्यूटेस यूज करते हैं। जब एक मॉलिक्यूल से दूसरे मॉलिक्यूल में फॉस्फेट ट्रांसफर करना हो तो हम काइनेस यूज करते हैं, तो हम काइनेस यूज करते हैं। सो आई हैव ऑलरेडी डिस्कस दिस इन डिटेल विद यू, राइट? सो जस्ट गिव मी अ थम्स अप बेटा, till now if the picture is clear to everyone, till now if the picture it is clear to everyone over here, yes? चलो जल्दी से एक थम्स अप दे दो, टिल नाउ इफ द पिक्चर इज क्लियर टू ईच एंड एवरीवन। यस, चिराग, जयनाम, वेरी गुड, वेरी गुड उस्मान, वेरी गुड बेटा, वेरी गुड हर्ष, वेरी गुड। चलो 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 चलो, नाउ ग्लूकोज 1 फॉस्फेट, ग्लूकोज 1 फॉस्फेट, yes, now this glucose 1 phosphate will combine with, okay, it will combine with UTP, right? It will combine with UTP, UTP. Now it is uridine triphosphate, uridine triphosphate. Now my question to everyone, uridine actually comes from uracil, and you guys will tell me कि uracil is present in RNA or uracil is present in DNA. चलो फटाफट से इसका भी आंसर दे दो, if my students knows it very well, so it is uridine triphosphate। चलो फटाफट से इसका आंसर दे दो, हाँ चलो फटाफट से इसका आंसर दे दो। चलो यस, चिराग ललवानी, वेरी गुड बेटा, वेरी गुड, वेरी गुड चिराग, उस्मान वेरी गुड, हर्ष, वरुण वेरी गुड, yes, uridine triphosphate। Now glucose 1 phosphate will combine with uridine triphosphate, okay, uridine triphosphate to form, okay, yeah, okay, chalo let me just write it nicely. They both will combine, yes, and what will they form? They will form UDP glucose. They will form UDP glucose. Now UDP basically means uridine diphosphate. Uridine diphosphate. Now when I say di, और यहाँ पे तो UTP था, UTP था, yes UTP था, तो कहने का मतलब है one phosphate is lost, one phosphate it is lost, and that is in the form of pyrophosphate, pyrophosphate, and this pyrophosphate is nothing but, yes, in short we write it as PPI, pyrophosphate। तो यूटीपी से एक फॉस्फेट चला गया तो क्या बन जाएगा, यूटीपी, यूटीपी बन जाएगा। और ये यूटीपी किससे कंबाइन कर रहा है, ग्लूकोज 1 फॉस्फेट से, तो ग्लूकोज 1 फॉस्फेट को आई हैव जस्ट रिटन ग्लूकोज ओवर हियर, I have just written glucose over here, so it is UDP glucose, it is UDP glucose, and the enzyme that is used for the synthesis over here, okay, of UDP glucose, remember beta, it is UDP glucopyro glucopyrophosphorylase, glucopyrophosphorylase, UDP glucopyrophosphorylase, UDP glucopyrophosphorylase। क्या इतना क्लियर है सब लोगों को? क्या इतना क्लियर है कि लास्ट में हमारा यूडीपी ग्लूकोज कैसे बन रहा है? यूडीपी ग्लूकोज कैसे बन रहा है? ग्लूकोज से ग्लूकोज 6 फॉस्फेट, ग्लूकोज 6 फॉस्फेट से ग्लूकोज 1 फॉस्फेट, ग्लूकोज 1 phosphate combines with UDP, uridine triphosphate. They both will combine, and yes, one phosphate will be removed from UTP, so it will be UDP, and this glucose will be like this. They both will combine, and this results in the formation of UDP-glucose. Yes, Chirag, Harsh, Jainam, Usman, very good बेटा. UDP-glucose. चलिए, इतना क्लियर हो गया? पहला स्टेप क्लियर हो गया, सिंथेसिस ऑफ UDP-glucose. I hope this is clear to everyone. Synthesis of UDP-glucose. Chal. Now coming on to the next step, beta. Coming on to the next step, that is action of glycogen synthase. Action of glycogen synthase. Now beta, you already know this glycogen synthase. Okay, it will act now. Let me just explain you over here. Okay, this will be, yeah, just understand this beta, what is happening now. Now you already know we have glycogen synthase. Okay, now this is the enzyme which is used. Okay, this is the rate limiting enzyme also. So yes, glycogen synthesis लिख दिया मैंने, glycogen synthase है, ठीक है. Now this enzyme cannot act directly. I have already told you this enzyme cannot act directly, so it needs a primer. Okay, it needs a primer, and you already know this primer. Okay, this is the primer, and this primer is nothing but, yes, it is containing glycogenin. Okay, this is right, containing this primer is glycogenin. Okay, it is a polypeptide which is made up of lot of tyrosine residues. Yes, so this is the primer beta. Now this primer is something on which this particular enzyme will act. Okay, this particular enzyme will act, right? Now you already know from the first step. From the first step, you are very well aware that we are having UDP glucose, uridine diphosphate glucose, and this UDP glucose can be multiple. Okay, एक तो है नहीं, बहुत सारे हो सकते, तो I can write n number of times. I can write n number of times because these are multiple, right? Now, what is the function of this glycogen synthase? Now, action of this enzyme is to transfer glucose. Okay, transfer glucose from UDP glucose to glycogenin, right? Now this glycogen synthesis is also creating a covalent bond, okay, which is actually alpha 1,4 linkage. Now before that, just remember beta, that glycogen synthase, I will write it over here, it is rate limiting enzyme of this reaction, of this cycle. Yes, it is the rate limiting enzyme. It is the rate limiting enzyme. It is the rate limiting enzyme. Now glycogen synthase का काम क्या है कि यूडीपी ग्लूकोज के साथ जो ग्लूकोज है, ठीक है, यूडीपी ग्लूकोज के साथ जो ग्लूकोज है, उस ग्लूकोज को यूडीपी से काट कर कहां ऐड कर देगा? ग्लाइकोजेनिन पे. कहां ऐड कर देगा? प्राइमर पे ऐड कर देगा. कहां ऐड कर देगा? प्राइमर पर ऐड कर देगा. So if we talk, what will be the product that will be formed beta? Yes, what will be the product that will be formed? Yes, remember over here, we have this glycogenin, right? We have this glycogenin, right? And yes, this is the primer. This is the primer. Yes, that is glycogenin. Now what will happen, beta? Over here, what will happen? Now the glucose molecules will be added. Now multiple glucose molecules will be added, and that glucose molecules I'm like showing with blue color. Okay, so यहाँ से पूरे सारे के सारे glucose molecules ऐड हो जाएंगे इस प्राइमर पर। ओके, सो यस, and you already know glycogen synthase, it is also creating a covalent bond. Okay, it is also creating a covalent bond because एक glucose दूसरे glucose से जुड़ेगा, which is why a bond that you already know, it is the alpha 1 to 4 linkage. Alpha 1 to 4 linkage. Yes. So I hope this is clear to each one of you. बेटा फटाफट से एक thumbs up दे दो कि second step समझ में आ गया है। Second step समझ में आ गया है। जल्दी से एक thumbs up दे दो कि if the second step is clear to everyone. हर्ष जयनाम, वेरी गुड बेटा। Usman, very good बेटा। So yes, now there can be more glucose molecules which can be added, right? Now the third step beta, the third step and the final step of this reaction, it is branching. It is branching, and remember the branching enzyme, it is used over here. Branching enzyme, it is used over here, and the name of the branching enzyme, it is alpha 1 4, and yes, alpha 1 4. Okay, let us write like this: alpha 1416 glucan transferase. Glucan transferase. So yes, yes, Usman, UDP is gone after detaching the glucose. Yes, yes, Usman, very right. UDP हट गया, ग्लूकोज जाके ग्लाइकोजेनिन पे ऐड हो गया, क्योंकि ये जो ग्लूकोज है ना बेटा, ये जो ग्लूकोज है, ये डायरेक्टली नहीं ऐड हो सकता। इट नीड समथिंग, इट नीड अ स्क्रैच, एक स्क्रैच पॉइंट चाहिए जहां से वो स्टार्ट हो सके। ठीक है, सो दैट्स व्हाई प्राइमर इज नीडेड। ओके, वेरी गुड स्मार्ट। चल, इतना क्लियर हो गया। अब इसका काम क्या होता है, अब इसका काम क्या होता है, इस ग्लूकन ट्रांसफरेज का काम होता है, यहाँ पे ध्यान से समझना है इस रिएक्शन को। लास्ट के जो 6 ग्लूकोज है बेटा, ओके, लास्ट के जो 6 ग्लूकोज है, 1 2 3 4 5 6, ओके, हेक्सासेक्राइड रेसिड्यू इज कट। इसको काटता है। ओके, 1 2 3 4 5 6 hexasaccharide residue is cut from the end. Okay, and this hexasaccharide residue, it is transferred to another point. Okay, that is called as a branching point, or it can be like this, can transfer to this point also. Okay, this point also. So remember beta, what will happen now, what will happen now, you already know that this is the primer part. This is the primer part, right? Now this primer contains, बेटा बुक के हिसाब से पूछ रहे हो द भाई फैक्ट, ये बुक के हिसाब से पूछ रहे हो या मेरे लेक्चर्स के हिसाब से पूछ रहे हो? जो पढ़ा रहा हूँ पहले वो तो पढ़ लो। मतलब मैं तो हैरान इस बात से हूँ कि मजे लेने के लिए तुम लोग को कुछ नहीं मिलता है, तो चलो लाइव सेशन में जाके टीचर से ही मजे ले लेते हैं। अभी मैं कुछ इंपॉर्टेंट बता रहा हूँ, तो उसमें अभी कौन सा चैप्टर है? अरे पूरे 40-50 मिनट हो गए लेक्चर को, और तुम पूछ रही हो कौन सा चैप्टर है। I would strongly recommend the Bhai fact to consult a psychiatrist after the session. सब ठीक हो जाएगा, हौसला रखो। ठीक है, now this primer, नाउ दिस प्राइम वन। अरे मतलब हद है, मतलब मैं आई एम जस्ट लाइक, क्या ही बोलूं मैं अब। अच्छे ऐसे ऐसे आते हैं, मतलब बेटा मेरा मेंटल पीस नहीं बिगाड़ पाओगे, हम डॉक्टर्स हैं। ठीक है ना, तो हमारे मेंटल पीस को बिल्कुल नहीं बिगाड़ पाओगे, हम इन चीज से बहुत ऊपर हैं। तो ये जो आते हो ना, ये सब हम करके छोड़ चुके हैं। ठीक है। देखो, ये होती है मतलब, इतनी ही हिम्मत है, अपने असली नाम से आओ, असली आईडी से आओ, फिर बताता हूँ मैं। अभी तो बेटा मुझे पता नहीं कौन सा चैप्टर है। ठीक है। चल। हाँ, कुछ ना कुछ ड्रामा होना चाहिए, ऐसे मजा नहीं आता ना बिल्कुल। तो ये देखो, ये कितने ग्लूकोज है, 1 2 3 4 5, तो ये 5 ग्लूकोज तो मैं यहाँ बना देता हूँ। ठीक है। राइट, और ये जो 6 कट के आए हैं, वो यहाँ ऐड हो जाएंगे। तो आई होप दिस इज क्लियर बेटा। आई होप दिस इज क्लियर। तो इसमें कुछ नहीं किया है, ब्रांचिंग में हमने जस्ट इसको दोबारा से आप बना सकते हो। एंड वी हैव जस्ट यूज दिस एंजाइम which is breaking the bond. Okay, yes, which is breaking the bond. और यहाँ पे जो लिंकेज देखने को मिलेगी बेटा, इट इज अल्फा 1 6 लिंकेज, अल्फा 1 6 लिंकेज, राइट। और यस, आपको पता है यहाँ से कौन सा बॉन्ड ब्रेक हुआ है, दैट इज अल्फा 1 4 लिंकेज, अल्फा 1 4 linkage. So that is something which is called as the branching enzyme beta, branching enzyme. So yes, this is the structure of the glycogen beta that you already get over here. Okay, this is the structure of the glycogen. It is in the form of a branching chain, right? So yes, in the muscle. I hope this is clear beta to everyone now. I hope this is clear how the glycogen is formed. Just I have shown you 3 steps and the glycogen is formed, right? I have shown you 3 steps so clearly, so visible on the screen. Jainam, very good. यस। अरे तुम ऐसे अजीबोगरीब नाम से क्यों आते हो, ग्लाइकोसिलाइटेड। ऑब्वियसली बात है, पॉलीपेप्टाइड है, टायरोसिन रेसिड्यूज है, वो ग्लाइकोसिलाइटेड ही होंगे ना। चल, ठीक है। हाँ, 1 4 बेटा, अब खुद देखो ना, इन दोनों ग्लूकोज के बीच में क्या प्रेजेंट होगा बेटा, इन दोनों ग्लूकोज के बीच में क्या प्रेजेंट होगा, 1 4 लिंकेज होगा। और 1 6 हमें कहाँ चाहिए, इस ब्रांचिंग पर चाहिए। ब्रांच को ऐड करने के लिए मैंने बताया था ना, 1 6 लिंकेज चाहिए। So that's why it is called alpha 1 4 1 6 glucan transferase. Okay, glucan transferase. Just by the name beta, just by the bonds that they are creating. Right? Alpha 1 6 branching पे होता है, और बाकी सारी linear chains में क्या होता है, alpha 1 4, alpha 1 4, right? So yes, Harsh, I hope it's clear to you. चलो, branching वाला हो गया बेटा, in the muscle as well as in the liver. Okay, चलो ये पॉइंट लिखिए सब लोग फटाफट से। in the in the muscle as well as, yes, in the muscle as well as in the liver. Remember, the glycogen, it is stored as beta particle. Okay, it is stored as beta particle, and one beta particle. Okay, remember, one beta particle, it is having 60,000 glucose residues. Okay, residues, and especially in case of muscles, right? One beta particle, it is having, yes, 60,000 glucose residues. 60,000 glucose residues. बेटा, अगर लिवर में बात करो ना, in liver specifically if we talk about, remember it is aggregates of beta particle. Okay, aggregates of beta particle, and that's why beta, aggregates of beta particle is located, right? Aggregates of beta particle is located, and that's why density is more in the liver. Density, it is more in the liver. Density, it is more in the liver. Beta, please ignore the comments of this terrorist. खुद ही terrorist ही लग रहा है वो. बताओ, इतने खाली है लोग, मतलब किसी के लाइव चैट में आके टाइम पास करना स्टार्ट कर देते हैं. मतलब हैट्स ऑफ टू यू बेटा, जो भी हो, बहुत अच्छा नाम कमा रहे हो भाई, घर वालों का अच्छा पैसा बर्बाद कर रहे हो. चल चल, let us now, I hope this is clear to everyone over here. ग्लाइकोजेनेसिस समझ में आ गया सब लोगों को? ग्लाइकोजेनेसिस समझ में आ गया? एक फटाफट से लास्ट थम्स अप दे दो, देन आई विल स्टार्ट विद ग्लाइकोजेनोलाइसिस. अभी जो हमने बनाया था उसको तोड़ के दिखाऊंगा. अभी जो हमने बनाया है उसको अब तोड़ के दिखाऊंगा, तो दैट इज ग्लाइकोजेनोलाइसिस. चलो हर्ष, वेरी गुड बेटा. जैनाम, ऐश, वेरी गुड. यस. उस्मान, चिराग, वेरी गुड बेटा. चलो let us now start. Let us now start with glycogenolysis. Glycogenolysis, right? Glycogenolysis. Now remember beta, if we talk about, okay, the concept I have already given you. Glycogenolysis में why body needs glycogenolysis? Now glycogenolysis beta, it will be active in which state? Fed state or fasting state, and under which hormone it will be active? So you already know that glycogenolysis means glycogen will be forming glucose. Okay, glycogen will be forming glucose. Okay, breakdown. Okay, breakdown of glycogen to form glucose. Yes, it is right. Jainam, Harsh, Usman, Chirag. Yes, Varun, very good beta. It is the fasting state. Yes, Varun Kashyap, it is glucagon now. Okay, you already know early fasting beta. When I say early fasting, you are very well aware that it is 4 to 16 hours after food. 4 to 16 hours after food. Okay, 4 to 16 hours after food. Yes. Yes, Varun, Harsh, very good beta. Now you already know 4 to 16 hours after food. Now, what will happen in this case? You are not having any food. Okay, 4 घंटे बाद अब ये तो है नहीं दोबारा खाने लग जाओगे. ठीक है, तो अब क्या होता है कि बॉडी में जो ग्लाइकोजन बना है दैट विल ब्रेक डाउन. ओके, एंड ब्रेक डाउन बेसिकली रिक्वायर्स अ हार्मोन दैट इज कॉल्ड एज hormone. Over here will be glucagon that will help in the breakdown of glycogen. Glucagon. Okay, yes, that is the hormone, right? So you already know hormone is glucagon, and obviously the liver glycogenolysis will take place. Okay, liver glycogenolysis will take place, right? And it is just 4 to 16 hours after food. 4 to 16 hours after food. Sure. Now again 2-3 questions beta that has been similar to every, okay, site. Okay, can anyone tell me the name or the organs which are like in which the glycogenolysis will take place? Yes, it is liver. Yes, it is muscle. जहां पे स्टोर हुआ था बेटा वहां तो होगा ही. Yes, it is liver, it is muscle. Yes, it is liver, it is muscle. And remember beta, yes, the organelle. Yes, Usman, very good beta. Organelle, it is cytoplasm. Okay, organelle, it is cytoplasm. And remember beta, 1 to 2% it will take place in the lysosomes also. Lysosomes are called as the dash of the cells. Can anyone tell me lysosomes are called as dash of the cells? Yes. 1 to 2% will take place in lysosomes. Yes. And yeah, we have lysosomal storage disorders also. We will be discussing about the Pompey's disease tomorrow, right? I will be covering the disease, glycogen storage disorders tomorrow. Okay. Metabolism पढ़ा है तो disorders भी पढ़ने हैं. We have to link it with clinical correlations, right? So yes, sure. Now let us now start with lysosomes are called as the suicidal bags of the cells, right? Sure. Let us now come on to the steps. Now, steps over here. Steps over here. Beta, there is firstly the action of glycogen phosphorylase. Action of glycogen phosphorylase. We will be talking about this, right? Secondly, the second step, it is removal of the branches. Removal of branches. And third step, beta. Third step will be conversion of glucose 1-phosphate to glucose even I don't have to tell you ये glucose 1-phosphate कहाँ present है बेटा खुद तुम लोगों को पता है खुद तुम लोग को पता है कि जब मैंने बात की थी ये glucose 1-phosphate okay this glucose 1-phosphate it is combining with UDP तो जब UDP ग्लूकोज बनता है बेटा तो ये फॉस्फेट ग्लूकोज के साथ एडेड है क्योंकि जो फॉस्फेट रिमूव हुआ था इट इज फ्रॉम द यूटीपी यूरिडीन ट्राइफॉस्फेट से फॉस्फेट रिमूव हुआ था ग्लूकोज 1-phosphate से ग्लूकोज 1-phosphate इज ऑलवेज एडेड ग्लूकोज 1-phosphate तो हमेशा से वो एडेड ही हो मैंने आगे लिखा नहीं है बट यू हैव दिस पिक्चर इन योर माइंड ग्लूकोज तो ग्लूकोज 1-phosphate एडेड ही है so it is like linked to one another in the form of glucose 1-phosphate only, right? So yes, now coming on to the first step, coming on to the first step, action of glycogen phosphorylase. Action of glycogen phosphorylase, right? Now what will happen, beta? Now let us start from where we already left, right? Let us start from where we already left. You already know we have this glycogenin, right? We have this glycogenin और ये क्या है हमारा प्राइमर है ये क्या है हमारा प्राइमर है और इसी ग्लाइकोजेनिन पर we have these glucose which are like added, right? We have the glucose which are added over here, right? So you already know, yeah, these are the glucose molecules which are added over this glycogenin primer, okay? Now beta what will happen now beta what will happen okay let us now yes count 1 2 3 4 5 6 now this glycogen phosphorylase what it will do beta now सबसे पहली बात इसकी दो तीन points you have to remember this glycogen phosphorylase basically it requires needs a vitamin okay it needs a coenzyme it needs a coenzyme to function and that is in the form of vitamin okay that is in the form of vitamin it is pyridoxal pyrophosphate okay sorry pyridoxal phosphate it is pyridoxal phosphate it is pyridoxal phosphate it is PLP pyridoxal phosphate okay so remember the highest content it is vitamin B6 beta vitamin B6 okay highest content okay the highest content of PLP it is in muscles highest content of PLP it is in muscles okay and approximately about 80%. Okay, it is approximately about 80% that you have to remember. Okay. Yes, 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 बेटा. हाँ बेटा, चिराग. ठीक है. Now what is happening? ये it's just giving a floor बेटा. ठीक है. आपको अगर मकान बनाना है तो उसके लिए क्या चाहिए होगा? बेसिक रिक्वायरमेंट क्या है? फ्लोर तो होगा ना. ठीक है. और मकान गिराना भी है तो वो फ्लोर तो वही रहेगा ना. फ्लोर तो कहीं चला नहीं जाएगा. कॉमन सेंस. ठीक है बेटा. मतलब it's the easiest way I can tell, right? Now चलो बेटा ध्यान से देखो. अब क्या होता है? Glycogen phosphorylase, it will cut the alpha 1,4 linkage one after the another. अब ये क्या करता है? one by one it will try to cut this alpha 1,4 linkage. अब पता है 5 और 6 कार्बन के बीच में क्या है? अल्फा. It is just a moment, let me just write it. Yeah, it is alpha 1,4 linkage. Alpha 1,4 linkage, you already know. So it will try to cut one by one only. Now this glycogen phosphorylase, it will cut, okay, the alpha 1 to 4 linkage, okay, one after another, one after another, okay, one by one after another, and from end से होगा. It will be from end beta. So you already know first ये वाला होगा, then yes, then what will happen? This will be out, this will be out, then this will be out. One by one all the glucose molecules will come out. Now this glucose beta which is coming out, this glucose which is coming out beta, this glucose which is coming out, remember beta, it is not in the form of single glucose, it is in the form of glucose 1 phosphate. ये ग्लूकोज के साथ फॉस्फेट लगा हुआ है बेटा. स्टार्टिंग में ही मैंने बताया था, यू हैव ऑलरेडी सीन दैट कि ग्लूकोज से ग्लूकोज 6 फॉस्फेट बनेगा, ग्लूकोज 6 फॉस्फेट से ग्लूकोज 1 फॉस्फेट, एंड ग्लूकोज 1 फॉस्फेट से यूटीपी कंबाइन करता है तो यूटीपी ग्लूकोज बन रहा है. बट दिस ग्लूकोज, ओके, इट इज ग्लूकोज 1 फॉस्फेट ओनली, क्योंकि फॉस्फेट ग्लूकोज से रिमूव नहीं हुआ, इट इज रिमूव फ्रॉम यूटीपी. Already I am not mentioned over here, but yeah, that is something that you have to keep in mind beta. It is glucose 1 phosphate will be coming out as the product, glucose 1 phosphate. Okay, now beta, now beta, remember beta, there is an end point for glycogen phosphorylase. ये glycogen phosphorylase का भी एक end point होता है. There is an end point for glycogen phosphorylase. Now this glycogen phosphorylase will only act, okay, it will only act up to a length of, it will only act up to a length of 4 glucose, it will only act up to a length of 4 glucose molecules, 4 glucose molecules from a branching point, from a branching point, and then it will stop its action, and then stop its action, and then stop its action. Yes, and then stop its action. So that is something which you have to remember beta. That is something which you have to remember कि ये सिर्फ 4 ग्लूकोज मॉलिक्यूल्स तक ही काम कर सकता है. तो रिमेंबर बेटा, ये 1 हो गया, 2 3 4, उसके बाद ये स्टॉप कर देगा एक्शन, उसके बाद इट विल स्टॉप दिस एक्शन. ओके, now coming on to the second part, the second reaction, that is, yes, that is the removal of branches, removal of branches, removal of branches. Now when I say removal of branches, it is by debranching enzyme, it is via debranching enzyme. Okay, it is via debranching enzyme, right? It is via debranching enzymes, right? Remember beta, debranching enzyme, it is a bifunctional enzyme. Okay, it is a bifunctional enzyme, means it has, yes. Now, you already know the enzymes are polypeptides. Now, when I say polypeptides, beta, when I say polypeptides, so yes, this is an enzyme which is having dual functions, beta. It will be having dual functions. So remember, yeah, one, it is alpha 1414 glucan transferase, glucan transferase, and this one, it is alpha 16 glucosidase. Okay, alpha 16 glucosidase. Now we will try to learn what are those, what is the action of these two enzymes? What is the action of these two enzymes? So let us now see beta what is happening. See beta, you already know this is the glycogenin. Okay, this is the glycogenin. Okay, this is glycogenin and this is the primer, right? This is the primer, right? Now to this glycogenin, basically glucose molecules are added. Okay, to this glycogenin, glucose molecules are added, right? Right. So yes, we have glucose molecules over here also, right? Now what will happen, beta? Now what will happen? Yeah, or I will just draw, yes, or remove this. Yes. Now that is the structure that we were left from. Now, what will happen, beta? Now, alpha 1414 glucan transferase. Now, remember, beta. Yes. Alpha 1414 glucan transferase. Glucan transferase. What it will do, beta? This glucan transferase will cut a trisaccharide. Okay, this will cut a trisaccharide. So yes, it will cut a trisaccharide. Yes, this. So three glucose units will come. So this is nothing but a trisaccharide. It is nothing but a trisaccharide. Yes, Rishiraj, namaskar, beta. Namaskar. So remember the alpha 1414 glucan transference. Basically, it cuts a trisaccharide. It cuts a trisaccharide. Okay, it cuts a trisaccharides. Okay, and you already know it is actually breaking one of the linkage over here, and that linkage beta you already know this linkage. It is alpha 14 linkage. It is alpha 14 linkage. Hence the name. It is alpha 1414 glucan transferase. Glucan transferase, right? Now, beta, you already know. Yes, you already know that we are now left with. Okay, we are now left with the glycogen. Okay, we are now left with the glycogenin. It is the primer, right? Glycogenin, that is the primer. And along with that, we have like these glucose molecules. Yes, we have these glucose molecules over here. Right, we have these glucose molecules and only one glucose molecules will be left over here. Okay, one glucose molecules क्योंकि तीन तो निकाल दिए तो एक ही बचा हुआ है. Now what will happen, beta? What will happen? This enzyme, okay, this enzyme that is alpha 16 glucosidase, alpha 16 glucosidase will cut this bond. Okay, it will help to cut this bond. Okay, and yes, it is breaking down which linkage, beta? Alpha 1 to 6 linkage, because branching point per what is there, beta? It is the alpha 16 linkage. branching point per what is there? It is the alpha 1,6 linkage, right? So now this glucose which is removed, okay, this glucose which is removed, beta, this glucose which is removed, remember, this is, yes, it is, yeah, it is removed as glucose 6 phosphate. Okay, it is removed as free glucose. Okay, it is removed as free glucose. Yeah. So this is the glucose which does not contain any phosphate. So this glucose removed, क्योंकि आपको पता ही है, yeah, it is not containing any phosphate. So that is something that you have to remember over here. Whenever the branching glucose is there, beta, that will not contain any phosphate group. So yes, it is removed as free glucose. It is removed as free glucose, beta. Okay, it is removed as free glucose. So yes, and we already know from our reaction we have glucose 1 phosphate. So glucose 1 phosphate can also be converted into glucose. So that is the third reaction that you have to remember. कि अगर we have direct cutting हो रही है branches की, if branches are directly cut from this branch or that branch also, what will happen? We have direct removal of this glucose 1 phosphate. Okay, तो ब्रांचिंग पॉइंट पर रिमूवल कैसे होता है, दैट आई हैव ऑलरेडी शोन यू. बट अगर डायरेक्ट हम लोग काट रहे हैं, सो हाउ this glucose 1 phosphate will get converted to glucose? So you have to remember that conversion of glucose 1 phosphate to glucose. Okay, glucose 1 phosphate to glucose. Peter, remember glucose 1 phosphate, it is the major product. क्योंकि फ्री ग्लूकोज तो सिर्फ ब्रांचिंग पे ही मिलेगा ना, वो तो एक ही ग्लूकोज मिलेगा. फ्री ग्लूकोज तो एक ही ग्लूकोज मिलेगा, ब्रांचेस तो मतलब हैं बट वो तो बहुत कम है एज कंपेयर टू ग्लूकोज 1 फॉस्फेट, क्योंकि एक एक बॉन्ड अगर ब्रेक करेंगे तो हमें बहुत सारे ग्लूकोज 1 फॉस्फेट मिलेंगे. तो मेजर प्रोडक्ट इज ग्लूकोज 1 फॉस्फेट. बट व्हाट वी विल डू विद ग्लूकोज 1 फॉस्फेट? बॉडी में तो ग्लूकोज चाहिए ना, बॉडी में तो ग्लूकोज चाहिए, राइट, बॉडी में तो ग्लूकोज चाहिए. सो नाउ व्हाट इज happening? What is happening, beta? Now this glucose 1 phosphate, okay, now this, okay, let us just understand this also. Glucose 1 phosphate, okay, it is differently metabolized in liver and muscle. Okay, differently metabolized in liver and muscle. Okay, how it is forming glucose now, we will learn this. How it is forming glucose now, let us learn this. Now you already know, for example, this is the liver. Okay, this is the liver. Okay, this is the liver. So remember, beta, what is happening? You already know we have, yes, glucose 1 phosphate. Okay, glucose 1 phosphate. Now this glucose 1 phosphate will convert into glucose 6 phosphate. Okay, it can convert into glucose 6 phosphate by the help of which enzyme, beta? Just name the enzyme, beta. It will convert into glucose 6 phosphate. Yes, from which enzyme, beta? Yes, mutase, very good. So this is phosphoglucomutase. It is very good, Janam, very good, Harsh. It is phosphoglucomutase. Okay, now this glucose 6 phosphate, it will convert into glucose with the help of enzyme phosphatase. Phosphate group remove करना है. You already know it is glucose 6 phosphatase. Glucose 6 phosphatase. Okay, so yes, it is removed as an inorganic phosphate, and a water molecule is also like released over here. So this glucose 6 phosphate, it is basically a hydrolase. Okay, it is a hydrolase, right? So yes, the water is required and inorganic phosphate is removed. Now, my question to everyone over here. 2 days back, I discussed that this particular enzyme, it is absent somewhere. Now, can anyone tell me this enzyme is absent somewhere? So, what is that place where this enzyme is absent? Where this enzyme is absent? चलो बताओ इसका आंसर बताओ, सबको पता है इसका आंसर, एवरीवन नोस दिस आंसर. या यस, वरुण कश्यप, हर्ष, उस्मान बेटा लिवर में तो है ही ना, लिवर में तो मैं दिखा ही रहा हूँ. ये मसल में प्रेजेंट नहीं होता. अगर सपोज मैं मसल ले लूं यहाँ पे, चलो ठीक है मसल भी ले लेते हैं, मसल के अंदर ये एंजाइम एब्सेंट होता है. यस, स्केलेटल मसल में. चलो फॉर एग्जांपल अब ये मैंने मसल ले लिया. नाउ यू ऑलरेडी नो वी हैव ग्लूकोज 1 फॉस्फेट. दिस इज मसल, स्केलेटल मसल्स ले लिए. ठीक है, यू ऑलरेडी नो वी हैव ग्लूकोज 1 फॉस्फेट. नाउ दिस ग्लूकोज 1 phosphate can convert into glucose 6 phosphate by the help of enzyme phosphoglucomutase. Phospho, yes, Usman Ash, very good beta. Phosphoglucomutase, you already know, but remember beta, in muscle we have absence of glucose 6 phosphatase. Glucose 6 phosphatase. Now beta tell me, will glucose 6 phosphate convert into glucose or not? Will it convert into glucose beta? Will it convert into glucose? Tell me beta. Will it convert into glucose? Yes or no? No. Chirag, very good beta. Remember beta, absence of this enzyme simply indicates glucose 6 phosphate, it cannot be converted to glucose. Glucose 6 phosphate cannot be converted to glucose. Yes, Chainam, Usman, Prince, Harsh, Ash, very good beta. It cannot be converted to glucose. Okay. Now, अब मसल में तो ये कंप्लीट ही नहीं हुआ. मसल में ग्लाइकोजेनोलाइसिस इज नॉट कंप्लीटेड. So remember beta, muscle glycogen is used when the muscle is exercising. Okay, so the glycogen is degraded and providing the ATP. अब ये glucose 6 phosphate, it will like directly undergo the process of glycolysis. Okay, it will directly undergo the process of glycolysis. It will not form glucose, right? It will not form glucose, right? It will not form glucose beta. Yes. So remember beta, the liver, if we say liver beta, if we say liver, so remember beta, the liver, it is a selfless organ. The liver, it is a selfless organ. Okay. It provides glucose to the blood. It provides glucose to the blood. It provides glucose to the blood. Whereas when we say muscle beta, muscle, it is a shellfish organ. Okay, it is a shellfish organ, and remember, it is utilizing the glucose or the glycogen for its own need. Okay, it is using glycogen or glucose for its own energy purpose. Obviously बात है कि अब खुद ध्यान दो कि ये ग्लूकोज 6 फॉस्फेट जो है, it cannot be converted to glucose. It will undergo aerobic glycolysis, and this aerobic glycolysis will provide ATP only to the muscle. It will provide energy only to the muscle. So that is why I'm saying that the muscle, is it is a shellfish organ, it is using the glycogen or the glucose 6 phosphate for its own energy purpose, right? So yes, now you already know in muscle, okay, what is happening whenever there is exercise, okay, heavy exercise beta, heavy exercise, what will happen? The muscle is exercising, so the muscle will have less blood supply. The muscle will have less blood supply, right? The muscle will have less blood supply. Okay, now what will happen? This glucose 6 phosphate will enter which glycolysis, guys? It will enter which glycolysis when there is less blood supply? I want answers from everyone in the chat box. If there is less blood supply, it will enter to which glycolysis? चलिए बताइए फटाफट से. Anaerobic, very good beta. It is anaerobic. It is anaerobic. Yeah, very good. चलो वेरी गुड. हर्ष, उस्मान, ऐश, चिराग, वेरी गुड बेटा, तुम सारे स्मार्ट हो गए हो. तो ग्लूकोज 6 फॉस्फेट एनएरोबिक ग्लाइकोलिसिस में जाएगा. अब मेरा सेकंड क्वेश्चन ये, एनएरोबिक ग्लाइकोलिसिस में नेट एटीपी गेन कितना होता है? एनएरोबिक ग्लाइकोलिसिस में टोटल एटीपी और नेट एटीपी गेन बताओ कितना होता है? चलो टोटल एटीपी और नेट एटीपी गेन बताओ कितना होता है? यू ऑलरेडी नो द आंसर, बट यहाँ पे थोड़ा सा डिफरेंट हो जाएगा. एनारोबिक में 10. हर्ष, मैं सही में जान ले लूंगा तुम्हारे अब. ओ गॉड. रिवीजन नहीं कर रहे हो बेटा? एनारोबिक में कितना ग्लाइकोलिसिस है? एनारोबिक में 4 एटीपीस मिलते हैं ना बेटा, 4 एटीपीस मिलते हैं. टोटल एटीपी 4 मिलते हैं. और नेट एटीपी, क्योंकि 2 एटीपी यूज हो जाते हैं, तो नेट एटीपी कितना हो जाता है? 2 एटीपीस. 2 एटीपीस. एन एरोबिक में बेटा तुम्हें पता है एनएडीएच से एटीपी नहीं मिलता. Ladita, thank you beta so much. Yes, it is 2 beta. It is 2 over here. But over here beta, when glucose 6 phosphate, it is converting into anaerobic, okay, it is undergoing anaerobic glycolysis. तो obviously बात है इसमें total ATP जो होगा, total ATP it will be 4, but net ATP will be 3. इसका रीजन बता सकता है कोई मुझे? इसका रीजन बता सकता है कोई मुझे? यहाँ पे नेट एटीपी 3 कैसे आ गया? अब मैंने आपको एनारोबिक में पढ़ाया था बेटा कि आपको पता था कि या एनारोबिक ग्लाइकोलाइसिस में ग्लूकोज से ग्लूकोज 6 फॉस्फेट बनता है, ग्लूकोज 6 फॉस्फेट से फ्रक्टोज 6 फॉस्फेट बनता है, और फिर बनता है फ्रक्टोज 1,6 बाइफॉस्फेट, राइट? तो एक एटीपी यहाँ यूज होता है, एक एटीपी यहाँ यूज होता है. तो एनारोबिक में 2 एटीपी यूज हो गए, तो 4 एटीपी बन रहे थे, तो 4 में से 2 गए, 2 ही एटीपी बचे. बट यहां पे जो हो रहा है, यहां पे डायरेक्ट रिएक्शन किससे स्टार्ट हो रही है? इट इज स्टार्टिंग फ्रॉम ग्लूकोज 6 फॉस्फेट. इट इज डायरेक्टली स्टार्टिंग फ्रॉम ग्लूकोज 6 फॉस्फेट. सो ये वाला रिएक्शन तो हुआ ही नहीं. तो इसमें एक ही एटीपी यूज हुई ना, इसमें एटीपी एक ही एटीपी यूज हुई. तो नेट एटीपी 4 माइनस 1, 4 माइनस 1 कितना हो जाएगा? 3 एटीपी. 3 ATP will be generated. 3 ATP will be generated. Is this clear बेटा? एक बार एनारोबिक ग्लाइकोलाइसिस पढ़ लेना, उसके बाद ये और अच्छे से क्लियर हो जाएगा. ओके, एक बार एनारोबिक ग्लाइकोलाइसिस पढ़ लेना और अच्छे से क्लियर हो जाएगा. ये समझ में आ गया सब लोगों को? चलो चलो, वेरी गुड. ऐश, उस्मान, वेरी गुड बेटा. चलो जैनम, वेरी गुड. Yeah, glucose 6 phosphate beta, it is present in the smooth endoplasmic reticulum. Okay. Yeah. एक और चीज बेटा, ये जो एंजाइम है ग्लूकोज 6 फॉस्फेटेज, ओके, ये मेंशन कर लेना. इट इज प्रेजेंट इन द एंडोप्लाज्मिक रेटिकुलम. इट इज द स्मूथ, इट इज प्रेजेंट इन द स्मूथ endoplasmic reticulum. Endoplasmic reticulum. And remember beta, second important point, that glucose 6 phosphatase, glucose 6 phosphatase, it is this enzyme, okay, this Yeah, I'll just write it nicely. So remember, beta, this enzyme, it is common to glycogenolysis. अभी हमने ये एंजाइम यूज किया है, ओके। और मैंने ग्लूकोनियोजेनेसिस में भी बताया था, मैंने ग्लूकोनियोजेनेसिस में भी बताया था आप लोगों को, राइट। So this enzyme, it is common to glycogenolysis and gluconeogenesis. मैंने एक क्लिनिकल केस भी कराया था आप लोगों को. I have discussed a clinical case with you guys when a child, when a newborn, it is, yeah, the yeah, it is the glycogenolysis and gluconeogenesis, it is not occurring in that newborn. So what is the enzyme which is deficient? What is the enzyme which is deficient in the newborn? It is glucose 6 phosphatase. That is how the clinical cases will be asked. That is how the clinical cases will be asked, right? So yes, so yes, you already know, I've already discussed one of the question also, glycogenin primer, it is glycosylated by the UDP glucose, right? Apart from that, glycogen phosphorylase, okay? What is the coenzyme for glycogen phosphorylase? Just comment in the chat box. What is the coenzyme for the glycogen phosphorylase? That is again a question that has been asked in the IMSS exam. Glycogen phosphorylase. Yeah, so vitamin, that is vitamin B6 pyridoxal pyrophosphate, right, beta? And remember that this enzyme, glycogen phosphorylase, it is the rate limiting enzyme. It is the rate limiting enzyme in glycogenolysis. It is the rate limiting enzyme in glycogenolysis, right? That I have already discussed. Yes, Chirag, very good, beta. Very good. So I have discussed everything. Yes, so let's end our discussion for today. So yes, so we have discussed about the glycogen metabolism today. Okay, we have discussed about the glycogen metabolism. Okay, so yes, just going through what we have discussed today about the glycogen beta, you already know some points regarding the glycogen and the bonds. Okay, the storage of glycogen in our body, it is basically in the liver and the muscle. Apart from that, the glycogen metabolism involves glycogenesis and glycogenolysis, right? And at what state they are taking place, the fed state and the fasting state, and which hormone it actually is responsible for this, the insulin and the glucagon. So glycogenesis, we have already talked about why glucose it is not stored in the cell, okay, why it is not in the actual structure, right? So yes, fed state I have already discussed, right? Or all the steps of the glycogenesis, glycogenolysis concept, site, organelles, steps, all I have discussed. So let's end the session for today. Goodbye. Goodbye.